We're now going to move into session 2 on medical reasons to develop medical nanobots, and it will give us an opportunity to explore the questions of this conference in a slightly different context. Our first talk is going to be by Dr. Russell Mumper, entitled Moving Toward Clinical Investigation with Pharmaceutically Engineered Lipid-Based Nanoparticles. Dr. Mumper is currently the John A. McNeil Distinguished Professor in the Division of Molecular Pharmaceutics in the School of Pharmacy, as well as the Director of the Center for Nanotechnology and Drug Delivery at the University of North Carolina at Chapel Hill. Dr. Mumper has more than 175 scientific publications and 30 patents or patents pending, and is also the co-founder of Pharmaceuticals, Inc. and Four Tigers, LLC. The title of Dr. Mumper's talk, Moving Toward Clinical Investigation with Pharmaceutically Engineered Lipid-Based Nanoparticles, really, I think, begins to bring us down to the hands-on, nitty-gritty, here and now of some of the very hopeful, fascinating, and positive theoretical structures and theoretical possibilities that were laid out in session 1. Russell, can you please join me at the podium? Thanks.
So I'd like to thank the organizers, and I was listening to some, I think, wonderful presentations this morning, kind of futuristic, out-of-the-box thinking, and I really appreciated that. And I couldn't help but think, well, okay, so now I'm going to talk about translational nanomedicine and what a damper when you start thinking about what has to be done to satisfy the Food and Drug Administration or regulatory body and getting a nanomedicine into a patient in the context of this morning's lectures. So I'm pressing the big red green button. I'm pushing it. And I'm waiting for something to happen. Okay. Am I doing that or you're doing that? Because I'm. Oh, okay. Okay. Am I able to advance it otherwise? Or should I just say next? Yes?
He's trying.
Nothing's happening.
Practical technology.
Yeah.
Yes, exactly.
Yeah. Okay. Oh, I had it. You're doing it, so I'll just nod to you when okay, so next slide, please. Okay, so what I wanted to do first is just spend one slide on kind of the realities of medicine, and particularly translational nanomedicine. So the work that my lab has been doing in the center that we've recently established is in the context of translational nanomedicine. So can we use nanotechnology in the near future, so in the time frame of 2 to 5 years, to address and solve clinical problems that are of relevance to human health? And you can't do that and not address what the Food and Drug Administration is thinking. So I'll mention that. And I mentioned that, so our work is translational in nature. So if you look at what this concept of nanotemplate engineering that we've developed is from starting with materials and their characterization to their interaction with cells, the blood components, what's the fate of these particles, and then can we address and solve particular problems? And our focus has been in addressing cancer and in potential new vaccine delivery systems. So I'll touch upon those. So the Food and Drug Administration has not yet established its own definition of nanotechnology. They are currently utilizing the definition as established by the National Nanotechnology Initiative. I think everyone was anticipating this report that was coming out last July 2007 on their interim nanotechnology report, and I think many of us that are interested in translational nanomedicine kind of read it with, I guess, a little bit of disappointment in that we expected much stronger guidance as to what would be needed. But the main take-home message was basically that we needed to improve scientific knowledge. And so that wasn't very comforting for us that are involved in submitting investigational new drug applications to the FDA utilizing nanotechnology developed in our labs or through our companies. And in fact, they're referring and specifically refer to a guidance document that was published in 2002 on liposome drug products. So liposomes have been around for 40 years. There's probably a couple of dozen maybe products that are utilizing liposome technology. And there's many pharmaceutical, pharmacokinetic issues that are involved with liposome delivery systems as cargo-based delivery systems, and they refer to that. Next slide, please. So what we've done is, in an effort to facilitate clinical investigation, after spending time in industry and working on colloidal systems and coacervates, we were looking for what we considered to be a scalable process to manufacture nanoparticles. And ideally, the lower left panel, the lower left part of the slide is a handbook of pharmaceutical excipients. And so if you page through this book of excipients, you'll come up with now hundreds of different excipients that are in various pharmaceutical products. Many of them are generally regarded as safe, so you hear this term GRAS. Different routes of administration, and in essence, the FDA has seen these type of excipients before. So what we sought to do this translational nanomedicine was to utilize materials already found in this book and to try to invent or establish some new way to make nanotechnology, nanoparticles in a scalable process, cost-effective, rapid, et cetera. And so what we developed is what we now call nanotemplate engineering, and it's a relatively simple process, although it took us 2 or 3 years to develop. But basically, the whole process utilizes a microemulsion precursor to make a solid particle or a particle that has a lipid component, a liquid lipid component on the inside. And specifically, these are warm oil and water microemulsion precursors. So we take a pharmaceutically acceptable matrix, solid at room temperature. It could be a fatty alcohol, fatty acid, fatty lipid, phospholipid, and you can melt it. And we'd like it to melt above body temperature. And once you melt it now you have your liquid oil phase, and that serves as a very good vehicle to dissolve many different types of drugs that we commonly are asked to formulate, poorly water insoluble or insoluble drugs. If you add that to warm water, or it could be a buffer or an isotonic sugar solution, it looks like warm milk. And so you have an emulsion-based system, very little stirring, very low mechanical stirring. And then simply by adding a surfactant, and the surfactant, and this is where the academic part becomes, I think, rich, you can immediately spontaneously form an oil and water microemulsion precursor. As these oil droplets, the interfacial surface tension goes very low. These systems go from warm milk to warm water, and then if you took out these systems, you can actually measure droplets in the size range analogous to a microemulsion, which are typically 5 to 140 nanometers. And as I mentioned, the interesting part is we can add many different types of surfactants to enable the surface properties of the particle that we ultimately desire. So these could be cationic, anionic, they could be reactive, pegylated, an antibody with PEG on the surface. And so if you do this right, you have what looks like warm water, and you simply let it cool. And so each of these oil droplets either solidify or encapsulate a liquid oil directly in that same vessel that you were using to manufacture the warm microemulsion. So literally in one vessel, in a period of 20 to 30 minutes, you can make these highly uniform monodisperse. I like to think of it as a natural way to make nanoparticles, because you're allowing the spontaneous formation of the microemulsion to do that. Next slide, please. And we've done a lot of things with them. I was just going to touch on a few of them. And all of these are either looking at their physical chemical properties, what they can or can't do. Obviously, if you're thinking about injectable products, your ability to make a sterile preparation, pyrogen-free, perhaps long storage, so you want these to be lyophilized. I'll talk about this in a minute. Biocompatibility, hemocompatibility is very important, and I'll talk about that in a few minutes. And then toxicity. And my colleague and I, who had developed this technology, have been looking at many different applications of this technology, and I'm going to touch on a couple of them today. Next slide. It's ready now? Okay. Okay, good. Okay, so conceptually then, this was a very potentially efficient and cost-effective way for us to practice nanotechnology. So microemulsions as precursors to solid nanoparticles. Because if you define the microemulsion window, basically the interface between the oil and surfactants and water used to form these systems, if you define the shape of this window, then it doesn't matter what the scale of these materials added, that if you can make this on a 1 mil scale or a 10 mil scale, then there's not too much reason why you couldn't do it at a 100-liter scale. And of course, for commercial manufacturing and clinical manufacturing, this is exactly what's needed. And some of the things that we've done in our lab is to show this. And so we've done this at a 5 mil scale, which is typical for our animal studies, all the way up to a 10-liter scale using conventional mixing. And we're able to form this microemulsion precursor within minutes and then cool the system in situ to form these 60 to 80 nanometer particles. One of the caveats with our system is we believe we need uniform heating and uniform cooling. So some type of jacketed system will probably be important. We have spent a fair amount of time doing what we call nanotoxicology. This is, in essence, because the FDA is eventually going to require this, but also because we believe that when you inject a nano system into the blood, what happens in the first few microseconds or seconds will dictate a lot of what the end result will be. So if it's activating or aggregating platelets, if it's lysing red blood cells, if it's coated with certain proteins and these particles then are directed someplace in the body, you could have a toxic event or you could certainly have lack of toxicity. And so the impetus for this work was to begin to address what happens when these systems are injected into the blood. And secondly, to be able to develop high throughput screening methods to understand, quote unquote, nanotoxicology of these systems. So fortunately, we're dealing with systems that have fatty alcohols and fatty acids, and we published a paper on that these systems could be metabolized by the body by alcohol dehydrogenase and NAD enzyme systems. We also published a paper in Pharmaceutical Research a few years ago that got some good recognition because it came out in a time, late 2005, where there had been several reports of the deleterious effects of various types of nanostructures in terms of their lack of hemocompatibility or toxicity. Of course, for establishing a proof of concept and getting into clinical studies, we needed to show negative effects of all of these. So we looked at red blood cell lysis, neutrophil activation, platelet activation and aggregation, whole blood clotting time, and what we concluded is that with the majority of these assays, we needed to approach somewhere between 500 to 1,000 or 1 milligram per ml, which would be the equivalent dose of about a 7-gram nanoparticle dose in the blood of a 70 kg human. And so, that work was well received in terms of establishing the methods, because as you know, if you've been involved with nanotoxicology, one of the concerns is a lack of suitable methodology to result in reproducible results from group to group. Okay, so I mentioned cancer and vaccines. That's where the majority of our work has been in our lab in trying to advance these to clinical testing. In particular, we have not been so much interested in cancer that is sensitive to a particular drug. Our work is largely focused on cancers, various cancers that are resistant. And there's many different mechanisms that cancer cells become resistant to otherwise efficacious cancer drugs. The one we've been looking at is multi-drug resistance mediated by P-glycoprotein. So it's an efflux system, energy-dependent, ATP-dependent, that causes otherwise effective drugs to be effluxed out of the cell. So either they're taken up to a lower degree or they're effluxed rapidly out. And we've seen in a number of different human cancer-resistant cell lines that we're able to package these drugs inside the nanoparticles, such as paclitaxel, doxorubicin, idarubicin, and others. And in cell lines, human cell lines that are overexpressing PGP, and otherwise you'd see micromolar IC50s of these drugs, that we're able to decrease that up to a log or even more. And we've translated these results in xenograft mice, so human cells in a xenograft nude mouse, or a syngeneic mouse model, so a mouse cell line in a mouse. And we've starting to understand the mechanisms. So we know that, and the data down here shows that the nanoparticles result in both increased uptake rates, so you're getting more drug into the cell, but you're also, the efflux rate is greatly reduced versus free drug. So influx rate is increased, the quantitative amount you get in, and then the efflux rate is reduced. So the overall effect is a significant reduction in the IC50. And we're now correlating that to PGP inhibition. So the components of these nanoparticles we are finding have two significant biological effects. One is that they inhibit PGP by mechanisms that we don't yet know. And second, they deplete the cell of ATP. So since PGP is ATP-dependent efflux mechanism, if you're depleting ATP, you're seeing decreased expression of PGP, you're getting more drug in, you're killing these cells. And we've extrapolated this to a couple of different models, and I'm going to just show you just a little bit of the data. Paclitaxel, so we take the anti-cancer drug paclitaxel in nanoparticles. If you take human glioblastoma cells and you compare the nanoparticle formulation to the commercially available Taxol formulation, and you look at the cell survival at 48 or 72 hours, they're comparable. This is a sensitive cell line, very low levels of PGP expression in this cell. So the drug can be entrapped and it can be released in the cell, and you see similar bioactivities. We can then take an adenocarcinoma, a colon cancer cell line, HCT15. It expresses very high levels of PGP, whereas the commercially available formulation, Taxol, you see very little effect on these cells, up approaching or over 1 micromolar concentrations. With the nanoparticles, you can significantly reduce that. And several groups have shown in vitro that with a colloidal type system you can achieve that. We then took it to the next step and said, well, what if we take those same cell lines, that's a human cell line, into a xenograft mouse, so a new mouse, and you establish subcutaneous tumors on the back. This is an extremely aggressive growing tumor. So you implant about 1 million cells, and within 19 days, these are about 3,000 millimeter cubed, approaching 2-gram tumors. And so these mice have to be sacrificed relatively quickly. And so if you inject, and you can see the injection of the paclitaxel nanoparticle formulation, you can significantly reduce the growth rate of these tumors. And we designed this study such that the cumulative dose of paclitaxel over this whole course of dosing was similar to a similar one-time injection of the commercially available Taxol product. We've also looked at a resistant mouse leukemia model. We used P388 ADR leukemia cells. So these are also cells, mouse leukemia cells that express very high levels of PGP. And you can establish a syngeneic mouse tumor model. If you inject these cells in these mice, they'll die within about 8 to 10 days, depending on the number of cells you administer. But if you take the doxorubicin nanoparticles, we actually looked at 2 different types versus control and even empty nanoparticles. You see an almost doubling of a lifetime of the mice. And so now we're doing obvious studies is that we're trying to extend this dosing out further to see if we can actually cure mice. People have shown that with the non-sensitive model, or with the sensitive model, with cumulative dosing of doxorubicin, you can cure these mice of leukemia, but this has not been shown for resistant leukemia. So we're encouraged by these results. Okay, so the last thing I wanted to mention was the other application that we're looking at is in the area of what I call nano-engineered vaccines, really trying to make viral mimetics, but without the potential hazards of using a virus for immunization. And so what we're doing is we're taking these nanoparticles and in essence we're trying to co-deliver both adjuvant and antigen to engineer an immune response that would be appropriate to either protect or cure a human of infection. And so there's many reasons to do that, so I won't belabor. But the main point is that if you can target these nanoparticles to potent immune cells, such as dendritic cells, you then may be able to orchestrate the immune response, either to get the appropriate cell-based immune response, which would be critical with a virus that was inside of a cell, or a humoral immune response, which may be appropriate for an extracellular pathogen. And these nanoparticles are taken up very readily by human dendritic cells, one of the most potent antigen-presenting cells. A couple of different things to show here. So we have positively charged nanoparticles, negatively charged nanoparticles, and neutral nanoparticles. And if you add them to bone marrow-derived dendritic cells, they're prolifically taken up. And it doesn't seem that charge has a big effect. These cells are highly phagocytic. The study or the panel at the right is a human dendritic cell. This was done with the Vaccine Research Center, a collaboration that we had. So one human dendritic cell, and you can see multiple endosomal compartments with some over 100 of our spherical nanoparticles, so rapidly taken up. And then to confirm that this is in fact a dendritic cell, and you can look at fluorescent nanoparticles throughout the whole cell, obviously not in the nucleus, but then you can stain with a fluorescently labeled antibody to confirm that it is in fact a dendritic cell. So rapidly taken up. And so over the past 6 or 7 years, we've been focused on an HIV vaccine. We started off doing a genetic immunization approach where we took a piece of plasmid DNA that we wanted to express conserved HIV proteins, but we're now using a subunit approach. And so our approach is to utilize conserved proteins, and we've been focusing in on TAT 1 through 72 and GAG P24. Importantly, these are both conserved, they're critical to the virus life cycle, and that there has been clinical results to show that long-term non-progressors, there is a relationship with CTLs, or cellular-based immune responses in long-term non-progressors. So our idea then is to use the nanoparticles to deliver these soluble proteins, look at novel ways to attach these proteins to increase the affinity of the nanoparticles, co-deliver a pattern recognition ligand or an adjuvant, target to dendritic cells to generate these potent antibody and cellular immune responses. And this is some data taken from 2 publications in Vaccine, 2004 and 2006. One thing that we recognize with these nanoparticles is that they're dose sparing. So if you imagined an HIV vaccine that you wanted to have multiple protein antigens, could be very expensive. The idea of conserving antigen and being able to get a response with 5 to 10-fold lower dose might be very important. So the panel on the left shows humoral immune responses, a dose response with nanoparticles, and aluminum hydroxide, which is the typical standard adjuvant that's approved in some vaccines, and compare that to complete Freud's adjuvant, an experimental toxic adjuvant, that you see a conserved immune response, total IgG with nanoparticles versus alum, where you rapidly see it fall off in the immune response. The other thing that we look at, and we do this many different ways, but one of the ways to get an indication of cellular immune response is to look at a ratio of 2 different IgG subtypes, that is the IgG2A and the IgG1 ratio. And with the nanoparticles, you can largely preserve that ratio to complete Freud's adjuvant, which is a well-known cellular immune response adjuvant, versus alum, which that ratio is very small. It's a Th2 or humoral immune response adjuvant. It's a very busy slide, but we also did epitope mapping. This is studies that we did with TAT 1 through 72. TAT is critical for inducing LTR transactivation, which causes viral replication. And so we were very interested in seeing what part of the TAT molecule were we generating immune responses to, and could it be different with nanoparticles compared to other methods of immunization. And so the main point here is that we detected very strong antibody responses to both this core region and basic domain region, which is involved with LTR transactivation and nuclear localization, and the N-terminal region. And certainly with alum, you lose this. With complete Freud's, it's a very strong adjuvant. You're able to retain that. And then we correlated that to loss of transactivation in an in vitro model in a dose-response way. So we're very encouraged by this. We originally set off to develop a prophylactic vaccine for HIV, we think that this strategy, having neutralizing antibodies to this specific region in TAT, may allow us to enter in clinical studies to test this as a therapeutic vaccine. And that is, since TAT is one of the first proteins, highly conserved, since it is expressed in the cell, essentially two things can happen. It can go extracellular, go in other cells, and then stimulate other cells to produce HIV, or in that same cell, it can transactivate that cell. So could we target this extracellular TAT? And so if you immunize in a therapeutic vaccine strategy, you develop neutralizing antibodies like I showed you on the previous slide. Those antibodies are produced. They will neutralize TAT, and specifically this critical region that causes LTR transactivation and nuclear localization. It neutralizes those antibodies. You can reduce HIV transactivation, maintain low viral bioburden. And could it be then that this could be a potential therapeutic vaccine for HIV to maybe adjuvant or replace the conventional heart therapy, which is expensive, has to be taken every day. And so this is something that we're looking at now. So all of what I told you in the last few minutes is, I think, made possible because of this, what we call the nanotemplate engineering. It's a way to make very reproducible nanoparticles, literally within 20 to 30 minutes, concentrations of 10 to the 14th per mil nanoparticles. And we're still focusing on many aspects of things that are needed to get things into clinical studies. Vaccines, many different advantages of these nanoparticle-based systems, from dose sparing to maintaining humoral immune responses, boosting cellular immune responses. And then in cancer, our work has been primarily focused on overcoming resistant cancer rather than focusing in on sensitive cancer. And I just want to end with lots of great conferences I know over the next 18 months. We've established 4 or 5 years ago a Chapel Hill Drug Conference, and in May 2009, more along the lines of this theme of translational nanomedicine, we are going to have a drug conference, and more news about this will be forthcoming. So thank you very much.